Okay, we're going to give everybody another minute or two to join, and then we'll get started. Okay, I think I'm just going to go ahead and get started. Yeah, so hi everyone. Welcome to our Invasive Species Center webinar. My name is Madeline, and I'm an Invasive Species Summer Technician and a student with the Invasive Species Center here in Sault Ste. Marie. I'm going to be the moderator for today, and the webinar today that we have titled is Investigating Starry Stonewort as an Ecosystem Engineer and Forecasting the Future Invasion Potential in Ontario Lakes. And so just before we begin, I would like to first mention that I'm speaking to you today from Sault Ste. Marie, and we are located in the Robinson-Huron Treaty Territory, the traditional territory of the Anishinaabe, home of Garden River First Nation, Batchewana First Nation, and the Métis Nation. And I would just like to extend my gratitude for their continuous stewardship of our surrounding land and water. Okay, so I'm just going to start off with a little bit about the Invasive Species Center. So the ISC is a not-for-profit organization that connects stakeholders, knowledge, and technology to prevent the introduction and spread of invasive species that harm Canada's environment, economy, and society. So we do have a lot of great invasive species resources on our website, including species profiles, best management practices, and more. So check us out at www.invasivespeciescenter.ca. And you can also sign up on our homepage for our newsletter, our bi-weekly media scan and event invitations, which is where you can hear about upcoming webinars just like this one. And before we start again, I'm just going to mention that if you do have any questions at all during the presentation, I'm just going to ask that you please type them into the chat box, and I'm going to read it to our presenter, Tyler, after the webinar. And if you're having any technical difficulties at any time, just please type them into the chat box or respond to the email found in your registration, and we're going to go ahead and resolve that for you. And we've also enabled closed captioning. So if you want to follow along that way, you can turn that on with the closed caption bar on your taskbar. And lastly, there's going to be a brief survey following this webinar. And if you could take some time to fill it out, we would really appreciate it. And so today we are welcoming Tyler. He is an expert speaker on Starry Stonewort. Tyler obtained a Bachelor of Science degree in Biological Science and a PhD in Applied Bioscience from Ontario Tech University in Oshawa, Ontario. Tyler's doctoral research investigated the role of Nyctalopsis obtusa, or Starry Stonewort, as an ecosystem engineer in Ontario lakes. Tyler is also a postdoctoral research associate at Toronto Metropolitan University, or formerly known as Ryerson University. Tyler's current research activities cover a range of topics, including invasive species, coastal wetland nutrient dynamics, and landscape scale modeling of contaminant fate. Tyler also actively participates in community-based environmental activities, and he's also currently a Director of Science, Research, and Monitoring with the Scugog Lake Stewards. So now I'm just going to go ahead and pass it on to Tyler to begin his presentation, and I'm just going to stop sharing my screen now.
Thanks, Madeline. Okay, can you see that okay? That's probably... Yes. Are you able to see the screen with the pointer okay?
Yep, we can see it.
Okay, so thanks for having me today. Today I'll provide just a brief overview for some of the research that Shelby, Dr. Andrea Kirkwood, and I have been conducting over the last 6 or so years, and where we investigated the distribution of starry stonewort in Ontario lakes and its role as an ecosystem engineer. So despite looking like an aquatic plant, starry stonewort is actually a green macroscopic alga, a part of the Ceraceae family. Ceraceae are described as stoneworts or brittleworts due to a marl incrustation on the thyli. Hopefully you can sort of see it here in panel B. Sometimes it'll have like a rough, rocky sort of appearance on the stem of the plant or alga here. The Ceraceae family includes 400 species worldwide, and many of them are now classified as threatened or critically endangered due to human-induced stressors. Starry stonewort is composed of a central thallus, so the main stem here, and at each of these little tiny internodal cells here, the little circles, branchlets will emerge in 5 to 8 per whorl. These branchlets can retain their shape when you remove them from water, unlike other members of the Ceraceae family. Starry stonewort has an asymmetrical branching pattern with brach cells separate from the internodal cells. So sometimes you'll see just one sort of branchlet coming off from a branch here, and it's not like diverging in the 5 to 8 as per usual. That's the brach cell. In comparison to other members of the Ceraceae family, starry stonewort often appears disheveled, as you can see here in panel C. And sexual organs can develop at any internodal cell. As you can see sort of here, these orange-red balls are male sexual reproductive organs. So far in North America, only the males or specimens that do not mature or have not matured have been observed. Starry stonewort has also been suggested to have the highest growth characteristic of all the Ceraceae, where it can grow from the sediment-water interface all the way up to the surface waters under ideal conditions, as you can see here in panel E. The star-shaped bulbul gives starry stonewort its common name, and this serves as an asexual starch body cell. These can be produced at any internodal cell, so where the branchlets emerge, but in Ontario specifically, most commonly these bulbils are found on the clear monofilament-like rhizoid or roots, as you can see in the panel here. The bulbul is useful for differentiating between Ceraceae. However, they can only be found within established beds, so self-sustaining, or from fragments that have been transported to new environments. So starry stonewort is native to Europe and Asia, stretching from the British Isles over to Japan. And in North America, the known distributions have been recorded for the central eastern portions, with the earliest record being observed in 1974. In Ontario, it was found in Lake Simcoe by Dr. Brian Ginn in 2008 and Presqu'île Bay of Lake Ontario by Dr. John Midwood in 2015. Currently, there's confirmed reports of starry stonewort in 2 provinces and 7 states. With an increasing awareness, anecdotal reports claiming widespread distribution across Ontario have started to emerge, like these 2 articles here. These claims have to be investigated, as starry stonewort has a history of misidentification both within the native and invaded regions, and Ontario houses abundant, diverse, vulnerable Ceraceae populations that are frequently misidentified as starry stonewort. So in this slide here, these are some examples of other Ceraceae members. So starry stonewort is commonly mistaken for the genera Tolipella, Cara, and Nutella. So as you can see from these images here, differentiating between Ceraceae can be tricky, not only because they look similar or have a similar appearance, but also because Ceraceae exhibit high degrees of phenotypic plasticity in varying environmental conditions. So phenotypic plasticity is the ability to change your appearance based on the environmental stimuli that you're inhabiting. So for example, if you could change the color of your eyes depending on the stimulus of light in your environment, where you would want blue eyes in low light and brown eyes in highlight environments, that's similar to what these alga species can do. So very few papers are available for starry stonewort biology, with the least understood mechanism of part of the invasion biology being dispersal. So generally, Ceraceae can undergo both asexual and sexual reproduction. And in Ceraceae, asexual reproduction can occur by fragmentation. So whether a little piece of the alga breaks off and is transported to a new environment, or by transport of the bobule, those little starch body cells, in Starry Stonewort's case, the ones that look like a star. These are vegetative fragments, and they're often called propagules. So dispersal and other charophytes are known to occur via wave action or the general flow of water within a given ecosystem for a relatively short period of time. The propagules can also be transported via organisms, whether they visit or feed on a charophyte meadow. And the most probable method discussed so far for transportation of Starry Stonewort within Ontario is transport on watercraft. So when a propagule is transported and deposited into a new environment or location, creation of a new algal bed is not instantaneous. Propagules will settle into the sediments, and when periods of low oxygen and low light transmittance occur at the sediment water interface, a new algal mat will start to grow. Sexual reproduction of charophytes or Curaceae is extremely rare and has been reported as such since the early 1800s. So for the most part, reproduction of Starry Stonewort within Ontario is thought to be occurring through asexual means. So in our research, we wanted to answer four main questions. First, we wanted to investigate the widespread distribution and determine how prevalent Starry Stonewort was actually across Ontario lakes. We also wanted to determine the physical and chemical habitat characteristics that constrain Starry Stonewort establishment within Ontario lakes. And with the literature being so scarce on potential effects on the environment and the associated communities within ecosystems that have been invaded, we wanted to determine how these ecosystem cycles and communities were affected once Starry Stonewort became established. And lastly, we wanted to determine whether Starry Stonewort was acting as an ecosystem engineer in invaded lakes of Ontario. So to be considered an ecosystem engineer, Starry Stonewort would have to have dramatic effects on the habitat structure and function. A good example of other ecosystem engineers would be beavers and how they can modify their habitat and all the cycles that are occurring there. So in Starry Stonewort's case, we thought that Starry Stonewort was altering two key lake ecosystem compartments, the sediments and the aquatic food web. So in order to address our research questions, we investigated five main objectives, sorry. The first was to evaluate the impact of Starry Stonewort invasion on the lower aquatic food web and habitat condition within Lake Scugog. So Starry Stonewort was likely introduced to Lake Scugog in the early 2000s, but it was first identified in 2015 by the Scugog Lake stewards. Lake Scugog served as a perfect sentinel or warning ecosystem to quantify potential effects on ecosystem processes and structure after Starry Stonewort invasion because it was a large, shallow, marl, so basic or rich in calcium carbonate macrophyte dominated headwater. In general, members of Curaceae are regarded as important components for shallow lake dynamics and stability all throughout Europe and Asia. So with Lake Scugog having confirmed Starry Stonewort populations throughout the lake, which have historically supported productive aquatic communities, Lake Scugog became a sentinel system for other lakes within Ontario. So Lake Scugog has had a history of invasive species. However, the system has always remained a macrophyte dominated ecosystem like you can see in these two images here. Although macrophyte dominated ecosystems can be a pain for recreational use, as you can hopefully see with the boat tracks through the Eurasian water milfoil beds in the left image, they typically support very healthy and abundant plant or productive ecosystems. The most notable invasive species to be introduced to Lake Scugog prior to Starry Stonewort was Eurasian water milfoil. Like you can see almost dense beds of Eurasian water milfoil on the right here. And Eurasian water milfoil was likely introduced to Lake Scugog in the 1970s, and it quickly dominated the macrophyte communities. Despite Eurasian water milfoil's presence, Lake Scugog continued to support productive and healthy ecosystems until around the time Starry Stonewort began to drastically increase. So Eurasian water milfoil has been termed the most aggressive aquatic plant to be introduced to North America, but with an increase of Starry Stonewort from 2016 to 2019, Eurasian water milfoil was displaced and largely constrained to one site within Lake Scugog. So in this image here, the yellow bar is Starry Stonewort and the gray bar is Eurasian water milfoil. And what we would also typically find is as Starry Stonewort would start to grow within a new site, it would wrap itself around Eurasian water milfoil stands until they fell over in the water column and died. And then the following month of sampling, they would just be completely replaced by Starry Stonewort here. So Starry Stonewort not only displaced Eurasian water milfoil, but had a consistent theme of displacement across communities of the lower aquatic food web, which are comprised of phytoplankton, macroinvertebrates, and macrophytes. So what's important to focus on in this image here is the shift in colors from the salmony color to the purple color, corresponding to the 4 years of study. So this shift indicates that the community composition was changing within Lake Scugog as Starry Stonewort became more prolific. This resulted in population collapses within the lower aquatic food web and decreased diversity across the lower aquatic food web. And typically diversity represents ecosystem health. And despite that, we still wanted to determine whether there was specific species interactions that were promoting the invasion of Starry Stonewort within Lake Scugog. So this plot here is looking at the top 10 most abundant taxa from the community comprising the lower aquatic food web. And what we can see in the boxes outlined in orange is Starry Stonewort generally had negative co-occurrence patterns with most organisms, which are indicated by the pink and purple little squares here. Starry Stonewort was found to positively co-occur with zebra mussels and also co-occur with chironomids or bloodworms, which are a macroinvertebrate or bug often used as a bioindicator for low oxygen concentrations. Starry Stonewort was also found to positively co-occur with Microcystis species, Microcystis, which is a harmful blue-green alga. And this coincided with our observations of emerging Microcystis blooms within Lake Scugog where no historical reports previously existed. So as our second research objective, we wanted to determine the role Starry Stonewort had in emerging Microcystis blooms within Lake Scugog. So despite Lake Scugog being a macrophyte dominated system, like you can see in the top image here, since its creation in the 1800s, with the establishment and subsequent increase of Starry Stonewort, periodic and prolonged Microcystis blooms began to emerge, as you can see in the bottom image here. With no previous historical reports, this was considered strange, as usually within macrophyte dominated ecosystems, the density of phytoplankton like Microcystis is kept low because there's competition for available space, light, and nutrients within the environment. Typically, phosphorus is a limiting nutrient for primary producers, and Lake Scugog has had a history of nutrient-rich waters, but with enhanced efforts, the phosphorus loading to Lake Scugog has been dramatically reduced in recent years. However, the positive association between Starry Stonewort and chironomids, the low oxygen indicator bugs, led us to question whether Starry Stonewort was affecting the cycling of oxygen, leading to internal loading of legacy phosphorus stored within the sediments, which would indirectly promote these emerging blooms here. So typically the bottom of productive lakes like Lake Scugog can have low oxygen concentrations by the midsummer, and that's because of the high biological oxygen demand. When oxygen is reduced at the sediment-water interface, conditions change, which allows inorganic phosphorus that was previously stored within the sediments to be re-released into the water column. So Lake Scugog frequently mixes just due to its orientation and prevailing winds and such, as well as the shallow mean depth. But because Starry Stonewort had such a high, dense growth characteristic, which typically formed dense benthic mats from the sediment-water interface to the surface waters, water column mixing was more subdued, and that allowed prolonged low oxygen conditions to occur, as we can see on the left outlined by the orange box here. It's also important to note that the low oxygen conditions occurred regardless of season, indicated by the 5 different colors here for each year of sampling. So other macrophytes, especially invasive species like Eurasian water milfoil, have been suggested to create the same observed events within environments. However, when we look at the 3 panels on the right here, where Starry Stonewort was not present and were dominated by Eurasian water milfoil, we often found that elevated oxygen conditions were present, indicating that the same observed events were not occurring due to Eurasian water milfoil. So the decrease of oxygen also appeared to respond strongly to increasing Starry Stonewort biomass. So here we have oxygen concentrations on the y-axis and Starry Stonewort on the x-axis. So due to the prolonged low oxygen conditions that we can see with Starry Stonewort presence, there was also a strong positive relationship between Starry Stonewort biomass and poor water soluble reactive phosphorus. So this indicated to us that because of the low oxygen conditions, the phosphorus or legacy phosphorus stored within the sediments was being remobilized into the water column. This is the main reason why we thought that it could be indirectly promoting Microcystis blooms as they emerged. So in order to identify parameters that were driving Microcystis bloom development within Lake Scugog, we used a statistical model called structural equation modeling. And what's important to note is variables included in this model were previously identified to contribute to Microcystis bloom development within other environments where they occur, like Lake Erie. We also included Starry Stonewort here, or obtusa, in order to test whether the observed positive co-occurrence between Microcystis and Starry Stonewort was serendipitous, or if there was some sort of biotic synergism occurring between these two problematic taxa. So what we can see here is climatic and other environmental parameters that have been known to contribute to Microcystis bloom development, such as precipitation and chloride, positively affected the presence of Microcystis species within Lake Scugog. And we also saw that total nitrogen was a constraining factor for these Microcystis bloom development within Lake Scugog. Interestingly, we only found that Starry Stonewort was the positive biotic or interaction between species within an environment contributing factor for this bloom development. And this was despite zebra mussels, so deep polymorpha here, being observed to contribute to Microcystis blooms in other systems such as Lake Erie. In Lake Scugog, zebra mussels were not a positive or significant factor for explaining these emerging blooms. So Lake Scugog was a good sentinel system to determine how Starry Stonewort may alter ecosystem structure and processes upon successful invasion, but we still wanted to map the distribution of Starry Stonewort in Ontario lakes and identify the habitat constraints using a subset of representative lakes. So as I mentioned previously, up until 2015, Starry Stonewort was only confirmed within Lake Simcoe and Presqu'île Bay of Lake Ontario. Within invaded regions of the United States, Starry Stonewort rapidly spread across 7 states. Ecological niche modeling from those regions identified calcium concentration as the main driving factor for successful establishment. And the primary source of calcium within aquatic ecosystems typically comes from underlying parent geology, mostly limestone, which in this image you can see in the southern portions of this map are the purpley color here. In Ontario, we have a unique geological transition zone called the land between. And within the land between, there's a south to north transition in underlying parent geology from a limestone to primarily granite with underlying or varying complexities, which will strongly influence the calcium concentrations within these lakes, as well as other important factors across this region. So with calcium concentration being the primary driving factor for successful establishment within the states, and with Starry Stonewort being identified to require a minimum calcium concentration between 15 and 25 milligrams per liter, the land between area offered an ideal naturally occurring study area to tease apart important habitat characteristics within Ontario. So we selected 60 candidate lakes from historical calcium concentration data across South Central Ontario. And over a 3-week period in 2019, we went to each candidate lake and sampled the Macrophyte community, as well as a long list of environmental parameters. So we were able to confirm Starry Stonewort at 37 sites within 19 of our candidate lakes. And Starry Stonewort was found to stretch from Brockville over to Eudora and north up into the Kawartha Lakes. 6 of the environmental parameters that we investigated significantly differed whether Starry Stonewort presence was detected. So typically where we would find Starry Stonewort, there were elevated concentrations of calcium, magnesium, potassium, and sodium. In addition, Starry Stonewort was often identified within shallow habitats closer to boat launch areas. However, as you can see from all the little dots in these plots here, there's considerable variation and overlap between whether Starry Stonewort was present or not. So in order to identify the habitat characteristics that either promote or constrain starry stonewort presence across Ontario, we developed an ecological niche model using a BRT or boosted regression tree approach. So what this model revealed was that the parameters site depth and the cations potassium and magnesium had the highest relative influence on starry stonewort presence across Ontario. Calcium was still an essential requirement for starry stonewort presence within an environment, but these parameters here were identified to have a higher promoting factor on the establishment. The ecological niche model also demonstrated that low concentrations of manganese were conducive for starry stonewort presence. So where you're in environments where you had higher manganese concentrations, you would actually see starry stonewort die off. An important deliverable and factor that weighed into this model selection for this portion of our research was the capability to predict or forecast species distributions by using additional data sets. So for example, in Ontario, there's several openly available data sets across the province, like the Lake Partner Program, and that incorporates some of the parameters that were identified to positively contribute to starry stonewort presence within our ecological niche model. So by applying the forecasting component of the BRT to those data sets, we would be able to provide lake managers and future researchers better insight in order to identify habitats that may be susceptible to starry stonewort invasion, which we'll talk about just in a short little bit. So next, we wanted to determine the spatial and environmental factors that influence starry stonewort's effects on the macrophyte communities across Ontario. So the complexity of Ontario's landscape influenced the community composition significantly across our 60 candidate lakes. Regardless, if we focus on the 19 lakes with starry stonewort presence, we start to see some interesting trends. So typically when we would find starry stonewort comprising more than 50% of the macrophyte community within a given lake, there tended to be lower macrophyte diversity. And in contrast, when we found starry stonewort to be less than 50% of the macrophyte community, there tended to be higher macrophyte diversity. Even though this figure here highlights the complexity and various lake characteristics that structure macrophyte communities across Ontario, I believe this also illustrates a very important feature of invasion biology that's largely overlooked. So typically when an invasive species is first documented or noted, primarily by drastic population increases, many think that the invasion must have just happened and we're seeing a population explosion. But in reality, it's quite common for the species to have established self-sustaining populations within the ecosystem, sometimes several years prior, and it's only being observed due to population increases following ecosystem disturbances, which give invasive species a competitive advantage due to their broad ecological tolerance. This, coupled with the fact that starry stonewort has a history of misidentification both within the native and invaded regions for decades, makes us believe that the establishment in lakes across Ontario was likely several years ago. So success of macrophytes, especially establishing invasive species, can have considerable spatial variation, and the extent that macrophytes can establish themselves is largely dependent on dispersal factors and the varying environmental conditions of any given habitat. So in this plot here, we wanted to investigate whether macrophyte communities across Ontario had influences based on land use and other important chemistry and physical characteristics for starry stonewort across Ontario. So what we can see is communities with starry stonewort outlined by blue here were commonly associated with elevated agricultural and developed land use within the lake's watershed. So these land use practices are known to create ecosystem stressors and disturbance, which could result in an alteration to the population dynamics in a given system, giving the invasive species of starry stonewort a competitive advantage due to the broad ecological tolerance. The data also support the notion that starry stonewort has and likely will continue to be misidentified within Ontario as observed across invaded regions of the States, as well as Europe and Asia. So here, if we focus on the orange box here, starry stonewort was found to highly co-occur with other native vulnerable members of the Caraceae family, which often lead to misidentification within native and invaded regions of the States. Comparative to the results observed within Lake Scugog, starry stonewort tended to have negative co-occurrence patterns with most other members of the macrophyte community, especially Eurasian water milfoil. Together, these results suggest that starry stonewort may be the most unrecognizable and aggressive macrophyte introduced into Ontario when disturbance allows starry stonewort to drastically increase its presence and effects. And finally, we wanted to apply our ecological niche model in order to forecast the distribution of starry stonewort across Ontario. So far, the majority of starry stonewort research and monitoring within Ontario has been conducted within the central eastern portions. And as you can see from this image here, Ontario is extremely hydrologically diverse, and it houses over 250,000 lakes. So in order to predict the potential invasional success of lakes across Ontario as a whole, we applied the forecasting component of our BRT model using openly available data from the Lake Partner Program. And this was done as a first preliminary step to start to investigate some surprising findings which came out of community monitoring efforts in 2021 now. So in this image here, we can see the majority of positive community monitoring reports came from environments where we have not been able to confirm starry stonewort presence previously, as well as these environments did not support conducive habitat characteristics for starry stonewort establishment within our water chemistry collection, as well as our ecological niche modeling previously. So primarily I'm focusing over here. So in order to create our forecasting components, we use three parameters that were identified within our ecological niche model to contribute to starry stonewort invasion. And these included calcium, Secchi depth, or a measure of how clear the water is, and total phosphorus concentrations. However, there were some spatially explicit data discrepancies between the openly available data. So in these images here, the LPP or Lake Partner Program data and the synoptic data, which is the data that we collected across Ontario. Regardless, our forecasting model that relied solely on calcium data performed the best, and this allowed us to create an accessible starting point to identify at-risk ecosystems for starry stonewort invasion. So here are three forecasting models that were produced using the openly available data, as well as our ecological niche model for starry stonewort across Ontario. And as a circle grows in size, as well as becomes darker, as you can see over here, that means that the probability of invasion within that lake is increasing. So here, again, we can see that the model which utilized just calcium concentrations performed the best. And this likely reflects the close linked relationship between starry stonewort and calcium, which is dated back to the 1800s. Now, if we zoom in on South Central Ontario lakes here, we can see the probability across this region. And surprisingly, not a lot of lakes seem to have a high probability of invasion across this region. However, our model can be improved with more spatially explicit data acquisition within shallow environments. These trends could reflect that discrepancy between available data and the ideal habitat conditions of starry stonewort. And with the increased data acquisition, we can refine this model to better predict where starry stonewort will become established. But as a starting point, we believe that this is a good entry level model. So given that our predictive model using calcium performed with a 97% efficacy or accuracy across the province, this may also reflect an increase in the vulnerable native charophytes in response to climate change, as has been documented in Europe and Asia. So this, coupled with anecdotal observations from our advisory roles of various community monitoring efforts, suggests that starry stonewort is actively being distributed across Ontario lakes by probably watercraft, and is almost always found to be entangled within increasing populations of the native vulnerable charophytes. So in conclusion, our results highlight that starry stonewort is often found in amongst other native vulnerable charophytes across Ontario, and this often confounds positive identifications, leading to an over-reported establishment than what currently has been confirmed in Ontario. Additionally, our research supports the notion that the majority of starry stonewort spread within Ontario is due to transport by watercraft, and currently management applications that are available for other aquatic macrophytes would not help reduce starry stonewort. They would likely exacerbate the negative effects of starry stonewort on the environment or remove vulnerable native macrophytes that are necessary to support Ontario's fish populations. Currently, our best course of action is to continue to collect spatially explicit data and increase education and awareness, especially surrounding proper clean, drain, and dry protocols to reduce the spread of Starry Stonewort and other aquatic invasive species in Ontario. So with that, I'd just like to thank all my colleagues from the Kirkwood Lab, the Scugog Lake Stewards, and our funding partners. And I'll just end on a funny little sciencey joke using the Nyctalopsis obtusa is Starry Stonewort's science name. So I'd be happy to take any questions.
Thank you, Tyler. That was a great presentation. It was very thorough and very informative. We do have a couple questions coming in. Michael says, you talked briefly about EWM and the mechanism for ecosystem change between it and Starry Stonewort. Is there any work done to differentiate whether EWM is a facilitator or a co-occur with Starry Stonewort?
Yeah, so some of our work from Lake Scugog and then across the 60 Ontario lakes, sorry, let me just pull it back up here, did sort of look at that question. So essentially, Eurasian water milfoil and Starry Stonewort are co-competers. They want the same habitat, they compete for the same environmental characteristics that would allow growth. And what we found was that Starry Stonewort actually outcompetes Eurasian water milfoil. Initially, they do co-occur. So within the early years of 2016, within Lake Scugog, you'd often find them side by side. But as Starry Stonewort became prominent and increased in abundance within those communities, Eurasian water milfoil was more constrained to one site that was heavily impacted by a golf course. And Starry Stonewort would just completely replace it across that from the Lake Scugog. And we saw similar results across Ontario where Starry Stonewort had a strong negative co-occurrence with Eurasian water milfoil. Let me just find the figure here. Sorry, this is, I think it's here. So in this figure here, M. spicatum is Eurasian water milfoil and N. obtusa Starry Stonewort. So again, we see a highly significant negative co-occurrence pattern between Eurasian water milfoil and Starry Stonewort, and that's across the 60 lakes in Ontario over quite a large geographical range.
Awesome. If there's any more questions, you guys can type them into the chat or the Q&A box. Colin says, can you recommend a technical charophyte guide?
Yeah, if you go into Google, there's one by Wood that was published in, I think it was 1962. And then there's like amended forum posts to that as charophytes, because most of them have been classified as extinct. However, in the context of climate change and DNA analysis and stuff like that, they're starting to find that they're actually not extinct, and they're updating some of their classification guides to adapt for more of the environmental changes that they've seen. But for the most part, across Ontario, I would really recommend the original Wood document with the amended forum posts.
Okay, Deborah has a question as well. She says, I own a cottage on West Twin Sister Lake, which is identified as having Starry Stonewort. We also have EWM and hybrid EWM confirmed by Dr. Sager of Trent. Our ratepayers group is putting together an adaptive management plan for EWM. This news about Starry Stonewort comes as news to us. We need help for developing a management plan. Who can help us?
In terms of management, current management applications for aquatic weeds would only exacerbate some of the negative effects exerted by Starry Stonewort. And especially what we saw across, so we actually sampled one of the twin sister lakes. I can't remember which one, I think it was West Twin. And what we found was that Starry Stonewort is actually, I'll just go here. Yeah, it was West Twin Sister Lake. So starry stonewort is there, but also native vulnerable charophyte populations that a lot of the fish communities depend on. So essentially, if you apply management applications that are currently out on the market without indirect or without targeted effects on starry stonewort, it's likely just to increase starry stonewort presence within your lake. And that's largely because, like, for instance, if you apply herbicide and such like that, you're priming the environment to allow those depositive propagules within the sediments to reproduce. I know some people have also used benthic mats and stuff like that, but benthic mats that are targeted for rooted. So charophytes don't have a root system. They have a rudimentary root system, but for the most part, if they're not actively generating a new algal mat, they're not associated with the sediments, and they can actually regulate their buoyancy. So stuff like that. So I know this is probably not what you want to hear, but for the most part, management, you should just leave it as it is and actually try and get some confirmation whether the entire population is starry stonewort or whether it's primarily dominated by the vulnerable charophytes. Because especially within the context of climate change across Europe and Asia, all other charophytes seem to be increasing. And that's leading to the re-examination of the conservation status. So you don't want to apply a management application and remove the vulnerable charophytes that have benefits and actually co-compete with starry stonewort to keep it sort of in check, if you will, and stuff like that. But if you'd like, there's a starry stonewort email I can send it out to you. Or if you want to reach out to me or Dr. Andrea Kirkwood, I'd be happy to discuss that with you further.
Okay, we have another question from Colin. Did your study area include Eastern Ontario, including Ottawa? I didn't quite see on the map earlier.
Yeah, so we went as far as Brockville, just because trying to sample the 60 lakes, multiple sites within a lake over a 3-week period proved difficult timing-wise. But I know some other researchers, I believe Dr. Andrea Kirkwood's working with them, may look at aquatic weeds in those sections. So maybe if you can connect with her. But I've also heard anecdotal reports that starry stonewort is prevalent around Ottawa. It has been found up stretching in the St. Lawrence River to northeastern Quebec. So I wouldn't be surprised if it's in that area in low abundance for the most part. But for our studies, we stopped at Brockville.
Okay. Michael says, thank you for your answer. Another question I was thinking about regarding the impacts that starry stonewort has in changing communities. How strong do you think the data is that starry stonewort is the driver and not the passenger of environmental changes? Is it unlikely that starry stonewort is only the beneficiary of co-occurring sources of environmental change that alter food webs, examples being agriculture and other invasive species?
Yeah, that's a great question. And that's part of the reason why we chose this. So general linear latent variable modeling is a new multivariate modeling approach where we can actually sort of tease apart those questions. So the strength of this modeling technique is using land use factors and other environmental covariates like you suggested here. You can actually maximize the mean variance relationship, remove those possible, influences, and actually get at whether or not starry stonewort is the causing factor here. The same with our structural equation model here. So by no means starry stonewort is the primary source of a lot of these changes, but it is a contributing source in addition to several other stressors that are causing these changes. And that was, again, primarily from Lake Scugog, but also some of our work that I presented here, as well as my other colleagues' work, is starting to show similar trends from Lake Scugog across the whole province as a whole. So I guess long-winded answer to your question, starry stonewort is not acting by itself to cause these changes, but it's definitely a driving factor that also needs to be considered.
Okay, Kevin asks, how would you characterize starry stonewort's impact as an invasive species relative to Phragmites' influence on our ecosystem? Is there a potential for starry stonewort to influence fish stocks?
Yeah, so Phragmites would mostly be like towards the common word, like the nearshore environment. Starry stonewort or charophytes in general have been shown to grow in depths upwards of 65 meters, depending on light transmittance and all that stuff. So I would expect starry stonewort to have a larger impact on fish communities, especially when it becomes so established and so dense that it starts affecting oxygen concentrations like we showed in Lake Scugog and stuff like that. Not only would it physically remove the possibility for fish spawning and refuge for other macroinvertebrates and food sources for the fish, but it would also alter like key chemical aspects that fish require. And Shelby started in some of Shelby's work, she's actually investigated that for Lake Scugog. So I would assume that starry stonewort would have a greater impact, especially when it becomes so dense and established that it's just typically a monoculture.
Okay, Deborah asks, we have been told no herbicide is effective against starry stonewort. We are planning to use Procelicor against the EWM. We need someone to help us identify how much starry stonewort we have, and is asking who can help us.
I'll put my email in the chat. If you want to email myself, Dr. Andrea Kirkwood's lab or myself can help you figure that out.
Perfect. Okay. David asks, are there any plans for citizen research beyond the original study, examples being via Friends of Algonquin, Friends of Killarney, to help identify the full extent of its spread?
I know there's been some discussions about community science monitoring efforts going forward. For this year, there isn't any, but we're always open to potential collaborations and research like that to increase education and data acquisition in the future. So again, if you just want to shoot me an email, I'd be happy to connect you with Andrea and myself, and we can talk about that.
Okay, and when will the link for the recorded webinar be available? I believe since we are recording this session, it's going to be uploaded to YouTube and can be accessed via our Invasive Species Center website. It should be up soon after the webinar, and yeah, we just have to upload it then. Okay, if there are any other questions, I'm going to give it just another minute here. Yeah. Do you see value in eDNA as a method for community science monitoring?
For starry stonewort specific or in general? Starry stonewort specific. Yes, although some of the more finer aspects of starry stonewort DNA need to be worked out a bit before that would actually work. We did collect DNA voucher specimens from all of the 60 lakes where it was at, or across the 60 lakes wherever we identified it, as well as all other charophytes. And we were trying to do some preliminary DNA analysis to look at whether there was different ecotypes. What we quickly learned is that a lot of the primers and stuff that are available currently don't work for these charophytes. But with refinement of the protocol and such like that, I think eDNA is a great tool, and hopefully it can be applied for this in the future.
Awesome. Yep. Perfect. So I think that looks like it might be it for the questions. I'm just going to go ahead and share my screen and just give a final spiel. Okay. So I just want to say thank you again for this great presentation. Thank you to everyone who tuned in this morning. The webinar has been recorded and it's going to be posted to our Invasive Species Center YouTube channel, so you can access it at any time. I'm sorry. All right, so we're also asking, just to reiterate, if you could fill out our ISC webinar series survey on our website. This helps our organization a lot on improving our presentations. And yeah, just to finish off, I just want to lastly give some attention to our next upcoming webinar about July reproduction in invasive forest insects, where we will have a guest expert speaker, Josephine Kwefalik. And this is going to be held on July 21st, and you're going to be able to register for this event and webinar soon on our website, just like you did for this one. So thank you, everyone. Thank you, Tyler.
Thanks for having me.
Wonderful. Okay. I think we're going to sign off now. Okay. Take care, everyone. Have a good rest of your day.